This look through the prism may start a fever, or perhaps prevent one. In 2010, chemists in the United Kingdom and India developed a simple method for fusing gold nanoparticles into an antibiotic. They found that the particles added to the stability of the molecules and might be useful in attacking some types of bacterial resistance. Now, a novel method of developing a vaccine that uses tiny gold particles has been designed to mimic a virus and carry specific proteins to the body's specialist immune cells. Here to look at whether a gold rush is the next big advance in medicine is Dr. James Crow. He's a professor of pediatrics, pathology, microbiology, and immunology at Vanderbilt University. Welcome, Dr. Crow.
Great. Thank you for having me.
Well, let's start with the first question, the obvious question. Why gold?
Well, people have made nanoparticles generally from metals, and there's all sorts of metals that can be manipulated. Cadmium, zinc, and gold is one of those. I'm a physician, and I'm really targeting things that can be ultimately put into humans. And I wanted to pick a material that would not meet a lot of obstacles in terms of safety. And we already knew that gold is injected into patients for some conditions like arthritis. So we knew it's safe from that standpoint, just the material gold. The actual gold that is currently licensed and used is a larger size, so what we're using is a smaller size. But if we had to pick a metal, we picked it based on the fact that we already knew we could inject patients with it.
And while gold may have some toxicity, it's certainly not nearly as toxic as something like cadmium, which as a former painter I know is not really the greatest substance to work with.
Right. Well, gold really has a high safety profile and record in medical use, so it's a good metal to start with.
Now, your method is very different in developing a vaccine. Usually we take a dead virus and we incorporate a little bit of it into the new medicine. What's your approach?
There are many ways to make vaccines. Historically, the ones that were developed in the 50s and 60s were live viruses that were weakened viruses, and you would take them and basically get an infection but no symptoms, and then you would get immunity. And that has been the central thrust of how to make vaccines for pneumonia like we're working on. But the problem is the virus that we're targeting is a virus that infects very young children, even in the first weeks of life. And it's been very difficult to come up with live viruses that are both safe and work in these young kids. So we're choosing a path to develop something that doesn't grow. It's not a living virus. We want to mimic a living virus. So that was part of the idea, to create something on the gold particles that look like a virus but is not living and doesn't have the potential to get out of hand in the young children.
And you mentioned the virus, so we should go into that a little bit. This is a respiratory virus. Why this virus? And can you tell us how bad this virus is, what this virus actually does?
Well, the virus is called respiratory syncytial virus, so it's a respiratory virus that causes wheezing and pneumonia, and it's actually the most common cause of wheezing and pneumonia in children throughout the world. It's very common. It causes hospitalization of children at a rate of about 1 in 100 to 1 in 200. So many of us know friends, neighbor, and family who've had a kid in the hospital with pneumonia with this. The wheezing and pneumonia can be severe enough to put kids on the ventilator. So every children's hospital in the country usually has 20 or 30 kids on a ventilator during the winter with this virus, so it can be very severe. It's also a problem in the elderly who have weakened immune systems, and about 17,000 elderly folks in the United States die of the virus every year. So it's a major target for vaccine development.
Now, 17,000 a year sounds like a fairly large, robust number. But overall, is this condition controlled? Are you building a better mousetrap, or are you building something that really is going to fill a need?
Well, currently there's no specific medicines for this virus, and there's no vaccines that have been licensed. So there's very little we can do about it right now. Basically, we put kids in the hospital, and we give them oxygen, and we give them IV fluids so they don't have to eat, and that's about all we can do right now, which is pretty crude given today's medicine. So we want to prevent the disease completely.
And so how will this vaccine do that?
Well, we have figured out a way to take the protein that is on the surface of the virus, which is what your body would normally encounter, and we've made that protein in the lab and then coated the gold particles with that protein. And the particles are approximately the same size as the naturally occurring virus. So we believe that these particles mimic the presentation of a normal virus. And the idea is then to use these as in a vaccine to expose children or elderly or whatever the target population is to the particles, and then their immune system would react to the protein thinking it was a virus, but in a safe way.
So the vaccine's going to stimulate a larger immune response because it's telling the body, hey, we are sick, before the body would naturally recognize that?
Right. The vaccine would present the protein and say, you have RSV in you, and the body's immune system would say, yeah, I see RSV, I need to mount an immune response. And mostly there are two components of the immune system. One is called antibodies, and the others are cells that are called T cells. And we showed that this vaccine reacts with both of those components of the immune system. So we think it'll kick off the immune system and induce protection.
So is the problem with RSV, is it really more about early enough identification, or are you just trying to say, like with the normal vaccine, let's make them a little bit sick or let the body think it's a little sick before there's anything whatsoever in order to get them well?
Well, what you're describing is treatment or prevention, and treatment is what drugs are for after you're sick. And what we're aiming for is to prevent the disease completely, so to fool the body to think the body's seen the infection without any disease ever occurring. So we're aiming to prevent the patient ever knowing that RSV was around.
Has this technique been incorporated successfully using other inorganic materials, or is this really novel?
People, to my knowledge, have never used gold in this way. There have been various types of particles in which people have encapsulated things. They're usually degradable particles. So the idea of putting a protein on a particle is not brand new. But I think what was conceptually new here is, first of all, using the gold, and also we developed techniques so we can control the shape of the gold. And we actually made the gold into little rods, and the virus is shaped like rods. So we're not only coating or embedding a particle, but we're simulating the geometry and shape of the virus, which we think is pretty novel, and we think it should work better than previous methods.
Now, you mentioned degradable, which puts into the mind, even though these are nanoparticles, so they're really, really small, what happens to the gold a month down the road, or a year down the road, or 5 years down the road?
Well, first of all, we're around nanoparticles all day, every day, that are naturally occurring. So walking over to the studio today, I was standing on the corner and a bus drove by, and diesel came out, and I breathed in a bunch of nanoparticles. So you're constantly exposed to naturally occurring nanoparticles, pollen, and a lot of things that are on the nano scale. So this is not unusual. And your body does have ways of clearing these particles. In our case, particularly in the respiratory tract, if we put this vaccine right into the airway, this is the type of thing you're exposed to all the time because you breathe in particles. And your body has a natural clearance mechanism with mucus and little cells that have little hair-like projections on them called cilia, and the cilia are constantly beating upwards so that the mucus will clear all the particles. So we would anticipate the body's immune system scavenger cells, which are called macrophages, would go and eat up the particles, and this cilia and mucus would eventually clear it in most cases.
And so in this case, it really wouldn't make a difference if the substance we're putting in is biodegradable or a metal. The body is designed to get rid of these very, very small particles.
That's right. And again, the history of using gold for arthritis shows that the body does deal with these materials. Their cells, the cells are called macrophages, and phage means to eat. So the cells move around and they eat things to help clear the body of foreign substances. So that's probably the main way that these sort of injected particles would be removed, that you have special cells that eat the particles anyway.
Now, this study is in a very early phase of testing, but just how early?
Well, we have fought through all the technical obstacles to making the protein, making the particles, formulating the vaccine. There were a lot of issues about the particles sticking together, and we had to work through formulations to make sure they were separate particles, which was our goal. And then we did studies to expose these particles to blood cells from healthy people. And this was kind of the exciting part, where we saw that the blood cells could process the vaccine and present it to other immune cells, and we saw an immune response in the lab using cells. So that's how far we've gotten. I think the next steps would be to move toward animal testing, and then if that looks good, move on to what are called phase 1, the early clinical trials. So the FDA requires animal testing as the next step. That would be what we would do next.
So at this stage, how successful was the vaccine? Were you having significant results?
Yeah, when we put the vaccine in essentially a plastic plate with active immune cells from healthy adults, the cells did recognize the vaccine, they picked it up, and then there are special cells called presenting cells, and these cells presented the vaccine to the cells we want to respond, the T cells in this case. And the T cells did respond, and the particular cells that were for the virus started expanding in the culture and growing as they should. So we demonstrated that human cells will process and respond to this. So that's very exciting.
And from this point to when it hits the shelves is one of those questions that you probably can't answer. But what is that process really like? What's a ballpark?
Well, the typical time span for vaccines in the United States has been something on the order of about 20 years from discovery to licensure and use in children, for instance. So there's a long road ahead for this type of thing. I think we need to test various doses and formulations in animals and get an idea how much do we need to use, how many times, can we just give it once, or do we need to give a second dose to boost and that sort of thing. So we've got some groundwork to do just on the strategy, and we also would look at some safety things in animals, and that usually takes a few years. Then we need to get publications out into the peer review process and have other people look at it, like the paper that we're describing, and let people comment and give us feedback. And so that whole process takes...
And replication as well, because you do want other labs trying this out, right?
Absolutely. I think we've told how to make these particles in the paper, and you'd like to see other labs do it. And I think another thing that we'd like to do is to use this for other viruses and see, is this a universal strategy that we can use for other viruses? Because the idea is we're mimicking the shape of viruses. Most viruses are on the nanoscale. So could we make viruses or virus-like particles to flu or HIV and other targets? So I think that's another direction we need to move in to see if we can replicate the findings but with other viruses.
And that's where I was going to go next, actually, the generalizability of it, because the implication of creating these inorganic viruses which would trigger a response and make someone immune from later infection or later susceptibility seems like it could be profound, could be very helpful.
Right. If we figure out the fundamental rules and principles about how to use these things effectively, we hope to use it as a platform technology where we just shuttle in different proteins so that the subsequent vaccine development projects go very rapidly because we learn from the first. And so we're trying to develop it as a general platform technology for all viruses. And one could even think about bacteria and fungi and potentially even cancer vaccines. There's a lot of applications for vaccines, but this is a start.
So probably the final question, and it goes back to it being gold. There are two things I think about with gold. One being that it's a relatively rare metal, and one, it's a relatively expensive metal. Would you like other scientists to also explore other metals as this progresses?
Well, I'm not sure what other metals would have a sufficient safety record to jumpstart into this area. I think there are other materials besides metals that one can formulate, and the whole field of nanotechnology is exploding over the last 10 or 15 years. So people have gotten to the point where you can write your name in atoms with some of these materials. So the ability to manipulate vast different kinds of materials is becoming available to us. So we might consider non-metal particles even biological materials that we could manipulate in the same way. So I think rather than a different metal, we would look for a different material altogether that could be manipulated in the same way by shape and size.
And the shape is really the key. So it could be gold, or it could be something organic. It could be maybe even a plastic?
Yes, it could be. I think our inclination would be to develop a biological material that could do that. We currently don't have that technology, but if we could mimic what the gold is doing in terms of building a shape atom by atom, that would be great. Now, the gold is interesting in that we can analyze the structure. For instance, there are very high energy microscopes where we can see individual gold atoms and how they pack into our particles. And some of the biological materials cannot even be analyzed that way. So that's kind of a next frontier of mimicking these nanoparticle structures with other materials. But we're good with gold right now. We think it's an exciting technology, and we're going to stick with it and try to further develop it.
Well, looking at the next step in 21st century goldsmithing, I'm Andrew Hiller for the Voice of Russia and the Prism, and I've been speaking with Dr. James Crow. He's a professor of pediatrics, pathology, microbiology, and immunology at Vanderbilt University. Thank you so much.
Great, thank you.